Everyone. Thank you for us to talk about what we've been working on for the past almost year. So my work has been or my group's work, I should say, has been exploring the effects of environmental drivers across ecological scales. So I want to start off by introducing our group. Um, Ali, Joey, Jeremy, Bethany, Evald, and myself, Sierra. We're all early career researchers at different stages of our PhDs or post grad school. So this work is really motivated by these broad signatures we're seeing of global change impacts across organisms and ecosystems. And one in particular that has been noted as this almost universal signature of warming is decreased body size with under climate change. However, when we look at different taxa, we see that some are responding negatively, some are responding positively, and some are seeing no change or ambiguous responses. So we can't always assume that organisms are going to respond the same way, which requires further work. And often because of just disciplinary divides, we think about the impacts of global changes at a single ecological scale. So physiologists think about fitness or body size changes in response to a global change driver. Population ecologists think about population declines. Community ecologists think about diversity loss. But really, the processes at a lower ecological level could be mediating dynamics at a higher ecological level in addition to the direct effects of these global change drivers. And using a trait-based perspective provides both a conceptual and a mechanistic framework for thinking about how fitness changes, in this case, body size, may mediate impacts at higher ecological levels, like at the population or community level. And whereas warming, as I introduced earlier, has received a lot of attention, here I'm showing literature reviews in fish species, other global change drivers have received less attention, and we often think about these single global change drivers in isolation. So it's really important to think about how they may be synergistically interacting to affect dynamics both at a single ecological level and across scales, which is kind of brings us to the questions motivating our work.
Okay, so as Sierra mentioned, those kind of key concepts is really what drove our project. And so here are two of our kind of big motivating questions. We did end up coming up with a couple, but these were just the main two to focus on for today. So the first being across systems, how are global change factors impacting individuals, populations, and communities? And then the second question being basically, are these effects correlated across these ecological levels of complexity? Now, it's kind of interesting to note for our motivating questions. You could maybe guess based on our illustrations that Sierra so beautifully made that our project mostly focuses on fish. Um, but it's kind of pertinent to know for our group that a lot of our time was spent actually thinking about what taxa we would apply these questions to, because they really can be applied to many different taxa. And so our project group for I'd say a good 2 months of how we started was very much on the team trees and plants versus birds versus fish. And so that's kind of how our project started out. So um, ultimately, kind of that drove our sort of data synthesis and harmonization, which is kind of what I'll go into. And so we ended up not working with the trees or plants, mostly, I think, as it was mentioned today. Um, just the idea that temporally, it would be very difficult to kind of answer the questions we were looking at because each of those ecological scales are changing at such different rates over time. So we didn't go with the trees. And then for the birds, kind of our main issue with this taxa was the data sets available don't have necessarily the individual fitness parameters that we need linked with abundance or population count. So there wasn't really any, or there were very few limited data sets that we could work with that included all of those ecological levels, which and then inevitably leaves us with the fish, which had some really great data. that included both individual size as well as population counts. And so after all that, those are kind of how we landed on fish some mere months later after the big fish versus birds debate that our project team went through. So those are, again, just kind of how we got there. And so for our data overview for where we're at for our projects, the main kind of criteria we were looking at was 5 years or more of co-located fish body size along with temperature and DO data. And then we ended up finding all of our sites are either neon or LTER for now, but it's comprised of different aquatic systems. So we have marine, lake, and stream sites. As far as our harmonization process goes, we didn't have too many filtering steps to work through, which was nice. We started first by mainly focusing on gear types to only use electrofishing or whatever the most common gear was at each site. And this helped kind of our calculations later. Then, for the most part, we just wanted to focus on observations that were, or I guess remove observations not identified at the species level and just to avoid bias in that case. And then we also removed any species that were found less than 3 years or sampling years at our sites. So what that left us with was 35 sampling sites. We had an average data set length of 20 years, which gave us some really fun data to work with around 431 unique fish species.
Awesome. Well, thanks, Ali. So Ali just described how we got this plentiful data set that we did. And what that looked like at first was just a huge data frame of fish catch. So as Ali mentioned, co-located data on fish size with our climate variables of DO and temp. So what we did next is to get us to a population level data set. We took that catch information and converted it to catch per unit effort, just the number of fish caught divided by whatever effort units it was, depending on gear type. And then we took that data set and calculated Shannon diversity to get us to a community level data set for our sites across time. So then we define these models, and we're working with hierarchical linear models for this project. And so these are going to they're going to enable us to estimate the sensitivities of these ecological components to our climate variables. And you'll notice, as I said, they're hierarchical because these intercepts are species and site specific. And then these sensitivities to DO and temp are also species and site specific. As a quick nod, so for example, in this individual model, our response variable size, we scaled by species and site. And then these climate variables, we scaled by site, such that they represent basically deviations from the mean of that particular site. And then for our other levels, our CPUE model looked really, really similar, again, of just intercepts that are species and site specific, and then responses that are species and site specific. And then for our community level data set, we just dropped these species specificities on these parameters because this response variable is at the whole community level. Okay, so when I say that these parameters are hierarchical, what I mean is we can represent these response parameters or these sensitivities as some kind of distribution that we're sampling from. So each species might get for example, a sensitivity to temperature that's drawn from one of these distributions. And in these hierarchical models, we are estimating these moments of the distribution as well, so the central tendency and the standard deviation. As a quick note, our original intent was to fit these huge, large hierarchical models that correspond to these models I have here. Due to computational issues that we're troubleshooting right now, that's not what we'll present today. Instead, what we have fit for now is a site-specific model. So basically this structure, but repeated at each site. So you drop these site terms, but it doesn't change too much of the interpretation that we'll show today. But we're working towards ultimately being able to aggregate these parameters across site by either troubleshooting and fitting that large overarching hierarchical model or using Bayesian meta-analysis approaches to aggregate those parameters. So today we're mainly going to focus on these mean effects from these distributions, which again is to say our best guess as to what the average sensitivity of the species are within a site of either size or CPUE. And this doesn't apply to this non-hierarchical term in the diversity metrics. These other parameters are really interesting to us, of course, like this variance term representing interspecific variation, I think is going to later on help us disaggregate some of the mechanisms. But for today, we're going to focus on some of these just mean effects. So to answer our first question, what we are going to show you today is these mean effects, or again, just the betas in terms of this diversity model across these different scales. So how are fish affected across these scales? We'll give you an idea of basically, are they generally positively affected, negatively affected, or if we're not detecting a lot of effects. And then for that second scaling question, what we're going to do is we're going to compare these parameters across these scales. So for example, we can plot these means, or in this individual versus population level effects, we can actually plot those species-specific responses as well to see whether or not they're corresponding with one another, and then bump up to the population and community level and ask that same sort of question. So now I'll pass it on to Joey to go through some of our preliminary results.
All right, thanks for setting us up for some results, Jeremy. So again, this is addressing the first question, which is just what is happening with these drivers at these different ecological scales. We're looking at an effect size plot. The grayed out points in the back are the non-significant ones, and we've bolded the significant ones just for clarity. The DO is the teal color, and the temperature is the tan color. So at our individual level, which is our body size metric, about 18% of sites responded positively to DO, and about 9% responded negatively. For temperature, about 10% of sites responded positively, and 7% responded negatively. And then as we go out to the population level, which is our catch per unit effort, which is just basically abundance metrics, 14% of sites responded positively to DO, and 5% responded negatively. And with temperature, we have 6% responding positively and 21% responding negatively. And lastly, going out to our community level, which is our diversity metrics, with DO, sites responded positively 14% of the time, and then 5% of the time responded negatively. And then temperature had 12% responding positively and 12% responding negatively. So now if we look at this information in bar graph form, we have our DO on the left and our temperature on the right, also looking at effects. The large gray portion of the bars are the non-significant sites. So you can do see that about 75% of the time we did not detect significant effects. And then in the orange were our detected negative effects. And in the fuchsia, purple, pink situation were the detected positive effects. From this, though, we can see there's few consistent effects of DO and temperature. But looking at DO, we tended to have a positive response, and at temperature, we tended to have more of a negative response. So if we're kind of zooming in on each of those three colors, the Morea Coral Reef was a great site where we actually have 3 sites at the site, all responding differently. So we have the back reef where it was, I'm trying to remember I'm not sure if this is DO or temperature, but we just see disparate responses based on where we're looking at on the reef. So we had the back reef responding negatively, the fringing reef responding positively, and the fore reef had a distribution pretty centered around 0. So a lot of species-specific responses, which can definitely inform the future direction that we're going to take with our analysis. And now getting into our second question, which is about the scaling of these effects. First, we're looking at the effects of individuals versus the effects of population. That dashed line is just a 1-to-1. So if they were perfectly matching each other, that's the kind of line we would expect. We can see that we do not see that, meaning that individual fitness responses are differing from the population size responses. And now if we're looking at our effective populations versus our effect on communities, again, we have that 1-to-1 black dashed line running through our figure. And we do see here that population size responses are similar to diversity responses.
Well, thank you for Joey for the next step. So now we are in the next step, and our team so thinks about like all the projects that can come out from that project. But now we focus on the manuscripts first, and we're working on it, and then also continue with our meeting, with our weekly meeting, and also we will meet with our project mentor to look for more input about that project. But we mainly focus on the manuscript, and then secondly, we'll be exploring mechanism underlying differences responses across ecological scale. So we think about within species, individual may differ, like interest, like thermal tolerance, like potentially influence populations level resistance. We also think within between species, like variations in life history or ecological role can drive divergence or compensatory responses, which potentially influence, shaping community structure, and also in terms of, system, like knowing like the marine system with high connectivity or also broad scale driver, in contrast to the other system, which is like very fragmented, the terrestrial system, which is fragmented, locally driven. So tend to exhibit stronger, adaptation, local adaptation. So from that, next slide, please. So we're thinking about like, so mainly as Ali mentioned previously in our presentation, so we use LTER and neon side. So to answer the question that we have in the previous slide, and then we're looking for data. So please, if anyone have more data, so please you can contact the teams or especially Bethany, so to coordinate with that. So our criteria are 5 years more, and also we're looking for data for the whole fish community. We're looking for temperature and DO data also. Next one. So at this stage, without like the significant contributions of our instructors, Marty, Nick Lyon, Carrie, so all of the staff, so we won't be at that point. Very, very significant contributions from that team. We also appreciate our mentor for his input and very significant input for now. We also appreciate all the team, all the CCAL fellows, so for the input they during our meeting. So I also appreciate my team. So for their flexibility, they are keep working even at now we keep working in data harmonizations. That we have such a very great team with different skill. So and also with that, I also want all the CCAT tonight helps you congratulate my teams. We have two new doctors in our team also, Dr. Jeremy and Dr. Sierra. And with that, so we're open for question.
Thank you, Evald. Thank you, full team. We've got a couple questions in the chat, I think, starting off from within the seeker group. So, Juliana, you can just unmute.
Hi, that was such a great talk, and your figures are so beautiful. And I really love your questions. I was wondering if you thought, like, it's interesting that there was like it seemed like a pretty even positive negative distribution of the effects of DO and temperature, and wondering if you considered an interaction between the two. I don't think about DO too much, but I was just thinking if metabolic demands are higher for fishes when it's hot, then the effects of DO may be particularly important, for instance, like when it's hot, or less important when it's cold or something like that.
Yeah, totally. Yes, there's probably tons of reasons to think that they are going to interact with one another. One thing, so interaction terms are definitely on our list. We've kind of got this like running list of things to try of more complicated model structures. Interaction terms are definitely there because, as you pointed out, Juliana, for sure, like the amount that temperature stress or DO stress is affecting is going to be contingent on the stress from this other thing. Um, Lag effects we're also kind of interested in as well. And then the big one right now, especially as the DO results relate to, is not using necessarily right now. These results are from last year's, I think, mean DO, and probably a lot of like major mortality effects or a lot of major things have to do with those mins and maxes. So we're going to look into using like lower quartiles or mins for DO going forward. But I totally agree that some of these like temperature stressors are probably contingent on DO and vice versa. So fingers crossed we have enough power to to fit that kind of model because I do think it's important.
Absolutely. Thanks. No.
Yeah, great job, team. I had a couple questions, one of which was more basic and the other one was slightly more involved. The first one was like, I just kind of probably missed it, but were you incorporating biomass or just density estimates for when you were looking at like your cash per unit effort?
Yeah, just density for now. We don't have biomass, but we do have length. So
I
we
mean,
could.
you can fit an allometric relation to that pretty easily for I think every fish in the LTER fish base does that pretty automatically.
Cool.
Um, the second question was like how you can incorporate fishing pressure or try to not incorporate fishing pressure is probably more likely because fishing often leads to size truncation, which is going to confound, especially at the individual level. It's going to make fish smaller. And as fishing pressure has increased over time, so too have temperatures and other factors. So you're probably getting a confounding effect of that. So I was wondering if you were able to not include that in some way, shape, or form, or how you were planning on dealing with that.
Yeah, that's a good question. That is also, not to be repetitive, also on our list. Specifically, actually, we got really lucky because our project mentor, Zach, one of the main issues, or I guess things we thought of only last week, was that some of our lake sites are stocked. And so related to fishing pressures. What we're considering now is for some of the lakes that have a larger proportion of our data. So I think one of our lakes is close to 50% stocked, kind of our path moving forward of either dropping sites and trying to find other data or, looking specifically at just dropping those species and working our way around that sort of bias that fishing or any sort of that human disturbance might create. So also on our list of things to look at. Yeah.
Yeah, I mean, if you assume that these effects are like species agnostic, it might just be easiest to remove certain species as opposed to removing whole data sets, so that way you can maintain as much
Yeah.
statistical
Well,
power.
and we've gotten really lucky too. I mean, on the plus side, with the LTR site in coordination with the DNR data, we have plenty of information on when they were stocked, which species were stocked, amounts that were stocked. So there's at least a feasible path forward. We just haven't gotten quite as far down yet.
Quick, thanks.
Awesome. And Byron.
Super nice work. Again, this is fantastic. And as I was listening to it, it occurred to me that there's some really interesting applications to climate change. And I wondered if you thought about how maybe interactive effects between temperature and DO would complicate some of these simple models people are promoting of climate change impacts and stuff like that, and how your work could inform or improve those approaches.
Sweet. Yeah, I can take that one. Yeah, totally. I think that yeah, Sierra made the point early in this intro that overwhelmingly a lot of these global change driver papers, especially as it relates to kind of these like fishery systems, look at one variable in particular, temperature specifically. And so one of the strengths is that, you know, bumping up to two variables, you can have like a lot of interesting interactions. So I think both the interactions between these two drivers as well as the kind of like interscale ideas, I think both create big cans of worms for actually forecasting these effects. But I think that this project will be a good first step if we are able to kind of like add in those layers of complexity into these, somewhat predictive models to be able to disentangle larger mechanisms of what actually is happening when you get into the murky waters of like, it's not just, a simple linear model with temperature. It's like much more complicated.